Well, that was the most timely thing. We never met. I was a curator that wrote it. I was the external reader under Bruce. And then after she went off for a postdoc, she sent me an email about having questions about it. Everywhere, no one will ever sit will sit in front. Showing.
You probably don't get to stand there right away. I think Mark's going to.
Yeah, well, he can stand there for a bit. I was just hanging on to something so I don't fall asleep or kill over.
Yeah, hi, good afternoon. So this should be an interesting talk because Josh has basically had 2 nights with virtually no sleep because of breaking down planes and things like that. So we're very lucky that he's here. So Josh is professor in the Ecology, Evolution, and Marine Biology Department at UCSB. His research sits at the interface of microbial and ecosystem ecology, as you'll see in a minute, and as well as his kind of work on drought and stress physiology. He also spends time in places like the Arctic, working out whether or not, as temperatures warm, we're likely to get a strong fertilization effect and pull a lot of carbon out of the atmosphere, or whether or not the microbes in the soil will beat the plants and we'll lose a lot of carbon to the atmosphere and get runaway climate change. So he's going to, I think, link out some of that broader world significance today within his talk. In my field, he's perhaps best known for some really highly influential reviews going from the early 90s to the 2000s to very recently. And a lot of the time when you think you've had an idea and you're really, really excited about it, you'll go back and find there's a Schimel publication from 95 where he's already told you to do it.
Or someone who wrote about it 20 years before that, too, as
As well as doing sort of the soil ecosystem ecology and the biogeochemistry. He's perhaps actually better known in many circles now for his book, Writing Science, which is really nice compared to most writing science books. In fact, it is actually really very pithy and actually draws on writer. If you want to buy a copy.
No.
The doctoral students will get more and more familiar tomorrow because there's a good number of people who are in a writing workshop with Josh tomorrow morning. We actually have like just a couple of spaces left in that writing workshop, which will be at 380 Edwards. So if you're interested, please email me and we can set you up with the getting started materials. But I'm not going to take more of your time.
Okay,
I'll let you get
thanks.
started. Thanks.
Well, it's sort of cliche to say I'm happy to be here, but today it's really true. Plane did break down in Santa Barbara and was 6 hours late, ultimately leaving. They had to bring a whole new plane down from San Francisco to get us up. So anyhow, I'm a soil scientist. That's what my PhD says. My job title says ecosystem ecology. And I live at that tension between, what am I and where am I and how does this all work? My undergraduate was in chemistry, and so I tend to think of this interface of micro and macro and how those scales relate. And I think that's where a lot of the really interesting science occurs. And so what I want to do in this talk is start with some of the more kind of conceptual framing of how I think about why dirt, why should people care about soil, and how do we think about it, and then move into some of the work that my group has been involved in where we're actually trying to apply some of that. So why soils? Other than I'm a soil scientist, so I think soils are cool, which for me is a perfectly valid reason, but it's not a very valid reason otherwise. When we look at the world system, all my friends at Santa Barbara who are marine scientists love, touting the ocean, but it's worth remembering that for all of the carbon that's in the ocean, mostly it's just bicarbonate, which is kind of boring from a biological point of view. That if you look at organic carbon in the ocean, there's less of it than there is in the atmosphere. And if you compare that to what's on land, Soil is the single big reservoir on the planet for potentially mobilizable carbon. So this is a very rough number. There's a lot in permafrost. There's a lot of uncertainty. But there is no question whatsoever that there's probably more organic carbon in soils than there is in the atmosphere as CO2, in the ocean as organic carbon, and in all of plant biomass combined. Soil's where the carbon's at. For this reason, of course, there's been a lot of interest in the idea of using soil as a way to mitigate climate change. The idea of the 4 per mill initiative, that if we can just increase global soil carbon stocks by 0.4% per year, that solves the problem. And that's a really clever number because it sounds small. It isn't really. But there are a lot of projects going on about ways of trying to sequester carbon in soil, which is a good place to sequester it. Because not only do you dump carbon there, but potentially increase plant growth. And so I'm working with some ranchers on the idea of you can get better cows and better soils, both. That's kind of a cool thing to do. The other reason soil is kind of becoming a major focus of people's interest these days is because of omics and the idea of the soil microbiome people think is wicked cool and very sexy, and all of a sudden people are very concerned about bacteria and fungi that live in the soil. And if you look at this paper by Janet Johnson and Kristen Hofmackel, the idea that the structure and the success of crops and of plants aren't just due to pathogens and nutrients, but actually the soil microbiome can influence the soil health. And so these two things, the importance of soil as a resource, people are realizing 95% or something of human calories come from soil. You know, we get some of our protein from fish, but the oceans are really biologically a desert. If you look at NPP, soil is where the world happens for humans. And this, just excitement over bugs and ohms. But if you do a metagenome or something on a soil, pick a gram of soil, you're likely to find something like 10,000 taxa of bacteria, add several thousand taxa of fungi, huge diversity, huge array of organisms, and this is really very interesting and very exciting to a lot of microbiologists. Not just microbiologists, but against that, I'll put the number of 3. There are 3 types of carbon in the classic century model, which is the dominant model that we have historically in the US used for understanding patterns of soil carbon dynamics. So if you look at century, there's fast carbon, slow carbon, and really slow carbon. That's kind of the way it thinks. It's based on 1980s understanding of soils. It looks very, very simplistic. This was developed by Bill Parton at Colorado State University. But Century can model the distribution of carbon across the Great Plains. India, in fact, has been involved in some really cool work with this in her past too, when she was not just being a dean, or I don't know if you're just being a dean, but it also is the tool we use for predicting soil carbon dynamics around the entire planet. The Earth system models that we used to describe climate are either based fundamentally on models that look like Century or Roth-C, the British equivalent of it, that think the same way. And they do that with only 3 flavors of carbon, and it's magic. It works remarkably well. If we look at Century, though, and think about how do we relate the way we conceptually think about and model soils, there are no different types of microbes in Century. It's not that there aren't microbes in Century. Bill Parton knew that a sterile soil would have decomposition rates of 0. Decomposition rates aren't 0. There is microbiology. It's not represented explicitly. Things happen and can be described without considering the ohms. The way these models think is they assume that microbes are in quasi-steady state equilibrium with their environment. And that means if you know the environmental conditions, you can predict the behavior of the microbes and then can model the process outcome. And you don't have to worry about the horrible, gory details of 10,000 groups of microorganisms in a gram of soil. We can predict their behavior well enough. essentially by just assigning response functions. If you want to know about microbial response to water, there's a nice simple relationship there. And again, these models work remarkably well. The one thing that Sentry really does know it has to represent more explicitly is not microbiology, it's the nature of the soil. Clay is fundamental in driving the behavior of these models, and soil texture is fundamental in driving the behavior of soil systems. So whether carbon sort of peaks out at 3 or 4.5 kilograms per meter squared is a function of clay. And a lot of the response functions in here include things like silt and clay content. You cannot model soil behavior without explicitly capturing something about texture. But of course, it's gotten worse than that. This is one of these normal things. The more we look, the more complicated it becomes. There's this recent paper by Craig Rasmussen et al. It came from a working group. I'm sort of 7th on the author list of that paper, but it's really Craig's work, that said, yeah, it's not even just clay that you have to worry about if you want to understand soil organic matter. You need to go beyond just clay. You need to think about clay mineralogy in more sophisticated ways to really capture the dynamics. And that's true, and we're getting there. And of course, Sentry also has 3 types of carbon, which are defined conceptually in terms of their turnover times. Well, is that what soil carbon really looks like? No, Sentry is a fantasy. The idea that there's carbon that's fast and slow. That describes reality, but that's not chemistry. As we actually look at soil organic matter, so if you look at a paper by Grandy and Neff, and you actually start doing meaningful analyses of what the molecules are in there, it's a huge array of discrete small molecules. The old ideas of humic acid, we don't even believe that molecule exists anymore in soil. These big convoluted condensed things. No, those are created in the sodium hydroxide extraction. In fact, soil organic matter is an array of small, discrete molecules that are stuck in places. So when and if you look at this classic paper by Marcus Kleber, that sort of pictures some of the interaction of how molecules may really structure on soils, yes, it's very convoluted and it varies depending on mineralogy as well. So microbiologists would love to see the microbes in the model because microbiologists say the processes that matter are carried out by microbes. Microbes rule. We want to see microbes in the model. Well, as soil scientists, we all know that minerals really rule the roost because physical structure in a physical environment is what dominates. And increasingly, we're coming to understand that organic matter in soil is not classes of compounds, it's discrete molecules that microbes have to deal with. Microbes have to make enzymes to deal with specific bonds and specific molecules, and if they can't afford to make that enzyme, they will leave that stuff on the side. There's no big airport in New Haven because it just isn't worth it for the airlines. You guys get your own way to the airport. It's the same thing for enzyme systems. You can get your own way here. I'm not putting on that enzyme. So soil is really very much a democracy in which the 3 political parties, this is a bad time to talk about this, of microbes, minerals, and molecules, are all in tension with each other, but they all play roles in regulating things. So, I'll sort of throw up this. It's the democracy of dirt. And I have a PhD in soil science, so I always say I'm entitled to call it dirt. Nobody else is, at least not for soil scientists. But now it's sort of that thinking about how we think about models, how we think about making them more complex, and enriching them. Do we need to? Sentry, as I will show, has its limits. That way of thinking has reached its boundaries. We need to go beyond that. And one of the major reasons for this is this idea of quasi-steady state, that you can predict the behavior of the systems based on the environment. And the world is increasingly non-quasi-steady state. Bill Parton and his colleagues helped really develop the model to describe patterns of carbon across the Great Plains where rainfall varies east to west, temperature varies north to south. It's pretty predictable. But the world isn't quasi-steady state. And if you look at the outside California, outside Santa Barbara, chaparral, grassland, gosh, there's even snow up in the mountains behind us in this photo. If I looked at my own backyard 10 years ago, right after it burned down, that was there. That's 30 feet from my house, well, maybe a little further, but the fire, we thought our house burned down that night. Or you look at the creek a year later after a flood. The world is non-steady state, and particularly in places like California, that's very true. But even on the East Coast, blow down in Wisconsin, Minnesota, flooding in Florida and the Carolinas, the idea that things are steady state is just a fiction. And models need to be able to address these perturbations and pulses, and we need to incorporate the questions of, so what do we need to understand about the systems to describe those? There's also, when we think about these kinds of biogeochemical models and the way they have framed our thinking, is there's very much a temperate system bias. Ecosystems, ecology kind of grew up in New England in forests, and it grew up in Colorado and places in grasslands, places with relatively predictable and well-behaved environments where, yeah, you can just draw a simple water response function and you won't be terribly wrong. But the world is actually a very dry place. Nice, moist, humid, predictable environments are the exception, probably not the rule. If you look at areas that classify out as arid, it's most of the US West, much of North Africa, Central Asia, down into South America, Australia, semi-arid, even wider ranges, Mediterranean systems. There are 4 of them, but they're small, but they make good wine and olive oil. And if you look at California, our seasons are all sort of backwards. I think Santa Barbara has 6 seasons, not 4, but we certainly have dry summers. It typically does not rain at all between March and hopefully October, November, sometimes not till January. But winter is the growing season. That's when it's wet. When we do get rains, grasses green up. And most of these areas are Spanish invasive grasses that were introduced with European colonization. So how do these kinds of our ways of thinking about biogeochemical systems as collapsed and expressed in models, how do they do in these kinds of environments where it isn't sort of constant, but where it's more pulsy? So a number of years ago, Tu Young Lee and several others of us worked on a project where he was trying to model using DayCent, which is a sort of updated version of the classic Century model, chaparral systems and looking at nitrate export from streams, so integrating to an ecosystem scale, and compare dry sort of pulsy years where you can see some points where they don't look like the bars like the predictions, versus some wetter, more constant years where the model results fit better. When we looked at nitrogen mineralization, net mineralization in measured model and how widely those differed across the seasons, particularly in the summer when rates are low, the model failed really quite horribly. So in terms of dealing with these kind of pulsy environments, DayCent, as good a model as it is, failed miserably because the assumptions that it makes are simply not valid in these environments. Models are tools for converting assumptions into conclusions, and that sounds critical, and it isn't really intended that way, but if the assumptions are wrong, the conclusions, of course, are equally wrong. So this kind of purely, let's assume quasi-steady state and assume we can model past the mechanisms, has reached its limit when we start moving to these more perturbed environments. We need to capture some mechanism more explicitly. The question is, what are the mechanisms? I mean, this picture suggests maybe it's microbes, and that's probably actually wrong, but how do we do that? What are the mechanisms that are key? And particularly, how do we do this in an environment where moisture is the real wild variable? And something that I've been really interested in for, well, really my entire career since I was introduced to microbial water dynamics in Mary Firestone's lab as a PhD student, is this idea of dry-wet cycles and something we still call the Birch effect, because Birch in 1958 published this paper where they noticed that if you take a soil and you dry it and re-wet it, you get a pulse of respiration. And you dry it and re-wet it, you get another pulse of respiration. You dry it and re-wet it, and each time you re-wet it, you get these bursts of respiration. And the question has remained, what causes those? And secondarily, how important are they in driving overall system function? So what causes the Birch effect? Well, one hypothesis had been, well, microbes die when things are dry, when they re-wet, and you get a release of microbial guts, and that fuels a pulse of growth in those that survive. That would ultimately sterilize the soil, and that doesn't happen. One thing that Mary was working on early on was this idea of osmolytes. Microbes live in water films, and they must equilibrate with the water potential of the soil around them. They cannot remain out of equilibrium unless they surround themselves in waxy coats and don't do anything. So either that means as water dries down and water potentials drop, they either dehydrate, or the thinking was they had to accumulate small mobile solutes to hold water within their cells, basically a simple osmotic effect. They needed to be as salty as it is effectively dry outside. But that means that when you suddenly zap them with a rainfall event, they either have to dump those chemicals very quickly, or water will flood into those cells and they will explode. And that is, in fact, what we thought happened for a long time, that there are suggestions that you could kill half the microbial biomass just by re-wetting a dry soil. I used to say that short of autoclaving, re-wetting a dry soil was the worst thing you could do to it. I don't believe that anymore. Most of my hypotheses turn out to be wrong, but we tend to learn some interesting stuff in that process. The third possibility, though, is that re-wetting mobilizes carbon and just fuels microbes with carbon that was soil carbon and now becomes microbial carbon. And so we've been working through some of these to try to understand them. So the first test we really did a mechanism, we in this case meaning Noah Fuhrer, it was part of his PhD work. So he did this experiment where he did essentially an isotope equilibration study where we added glucose to soil, radioactive glucose. My colleagues who work on radiocarbon hate me, but I love C14 as a tracer. It's cheap, it's very sensitive, it's really easy to use, but you can't go anywhere within 10 miles of an accelerator mass spec if you've even said the word. You incubate it into the microbial biomass, so we're basically labeling the biomass. Then we dried the soil down, so hopefully microbes would acclimate to those conditions. And then we re-wet the soils and measured the amount of CO2 and the enrichment of the CO2, the microbial biomass, and of the dissolved organic carbon, the carbon that's released into just soil solution as a result of re-wetting. And what we saw was, looking at two different soils, was this initial up to the re-wetting at day 10, you could see that the CO2 sort of equilibrated, but then when we re-wet, we got a spike where it was going way high. The microbial biomass as a whole had an enrichment of about 4. So yes, the enrichment of CO2 equilibrated with the biomass, but the re-wetting pulse was highly enriched relative to the average biomass, suggesting, yes, this is a small fraction of carbon from a small portion of the overall microbial population. The DOC that was mobilized by re-wetting was basically radioactively cold. So we're mobilizing unlabeled carbon, but we're aspiring labeled carbon. So this showed that this was microbial carbon that we're releasing in this first initial pulse, but it doesn't say what chemical form the stuff had been in, whether it was actually microbes. And we did see a big flush of DOC that was mobilized that was not immediately respired. So we hypothesized that as soils are drying, microbes will turn this stuff into osmolytes. Bacteria have classically been thought to use nitrogenous compounds, glutamate, proline, trimethylglycine, which always seem kind of stupid. Why would you use a resource so nitrogen-rich that you have to get rid of? But that's what pure culture shows. Fungi were thought mostly to use glycerol and mannitol, nitrogen-free. Increasingly, we're seeing evidence that trehalose, a glucose dimer, may play a role, and I'll show some data about that in a few minutes. In pure culture, these costs can be very large to grow bacteria in pure culture under salt stress. 30% of total cell carbon, as much as 60% of bacterial cell nitrogen can be tied up in osmolytes when they're growing under salt stress. So potentially a huge pool of carbon and nitrogen that would have to be released immediately. So Claudia Boot, when she was a postdoc in my lab, worked on looking at these. So we extract microbes, look at the chemicals in the microbial biomass. And this is just osmolyte carbon, just per gram soil. Glutamate was the one amino acid compound that was most prevalent. And you can see it sort of wobbles around. It's always at a fairly high number. In the summer of, well, that was actually now 2008. Wow, that was a long time ago. It's not behaving like you'd think an osmolyte would behave. It's high in the summer, but it should be increasing early in the summer as soil's dry, and that's not what we saw. And it's higher yet in the winter. When she looked at trimethylglycine, nothing there really at all to talk about. And proline, the classic gram-positive osmolyte that Ken Killam, when he worked with Mary Firestone, saw lots of, we didn't see any of. When we normalize these numbers to just total microbial biomass, and this just for glutamate, what we see though is if you look at the fraction of microbial carbon that's in glutamate, it's basically flat. Zero evidence, or actually evidence that these compounds are not accumulating in the microbial biomass. They're not using nitrogenous osmolytes in microbial biomass. All the pure culture work was just dead wrong when it applies to the field. Now, why would that be? Well, you know, cultures that don't accumulate proline won't grow under salt stress in a lab flask. They may survive in soil, and so, you know, if you do things in pure culture, you're seeing a very small fraction of irrelevant organisms potentially. But in the field, they were not doing that. Amy Miller, working in a sort of similar period, did experiments working with John Milak and others where she was interested in the effect of multiple cycles of pulsing. I showed you that Noah showed in a single pulse you get it's from microbes. Amy did an experiment where she either re-wet these soils every 2 weeks or every 4 weeks. And what she saw was that every time you re-wet a dry soil, you got a spike of respiration. But when you let things sit dry for 4 weeks instead of 2 weeks, the spikes were twice as big. So something was happening during the dry period that was mobilizing carbon, and I still don't understand what that is, but I'll talk about some of that. When we sort of compared, so is this a big number or a small number? Yeah, we get these spikes, but do they matter? She looked at how those relate to total respiration when soils were held under constant moisture, and up to about 60% of water holding capacity. That's sort of optimal. And both of those re-wetting treatments, they're plotted at their average across the entire period water content, respired a lot more carbon than even optimal constant moisture. That difference is about 1,000 micrograms of carbon per gram soil, and that's about how much carbon was in the microbial biomass. So that's a lot of carbon, actually. It's not a trivial number, and this is mobilized somehow by the drying and re-wetting process, and the length of period the soils were dry played a role in regulating that. So the pet hypothesis I came up with to explain this was that microbes require a certain amount of water to respire, but the extracellular enzymes they produce to break down plant detritus may require less water. And so that even though soils may dry and microbial activity may dry, perhaps the enzymes continue to work. And this was, I thought, a good hypothesis. Eric Slesserev, who is currently a PhD student in my lab, decided to take this on as part of a paper we've just published that Pete Homyak was lead on. Eric is just starting a postdoc, but when he comes around to applying for faculty jobs, he's like one of the best students I've ever had. You should hire him. If you could get him to come here. He did the study where we incubated plant roots dry and looked at the potential activities if we incubated them both with and without chloroform. So chloroform kills microbes, allows exoenzyme activity, so you can eliminate biological uptake of any reaction products. So what he found was that the enzyme potentials are there on those dry roots. There's lots of enzymes on those dead roots in these soils. But when he then looked at the actual product, how much of the products of these enzymes you could accumulate, they did not. The enzyme potentials are there, but they're not active. PET hypothesis was dead wrong. The enzymes seem to require water just as much as the microbes do. And so this accumulating carbon that we see during the dry periods is not from detrital breakdown. And we see this just in the field, in our field sites in California, that bioavailable carbon increases through the summer. So we thought it was from root breakdown, and we were wrong. And Eric showed it to us. And yet, we do know that mobilizing this carbon and drying and rewetting cycles not only increases microbial carbon release, it actually grows microbes, that we mobilize carbon. So this was work from Shirang Xiang, published in SBNB. In surface soils, we had initial soils that were incubated, constant moist soils that were wet and dry, I think 6 times. And we could increase biomass by, maybe 50% in surface soils. In deep soils, B-horizon soils, we were able to increase microbial biomass by a factor of 6. Now, in those soils, if you just wet them and leave them moist, respiration spikes and then drops to 0. But if you just keep drying and rewetting, you keep mobilizing carbon, we could grow a lot of microbes. So somehow, wetting is mobilizing carbon to microbes that's bioavailable, they just wouldn't otherwise see. And the paper that Martin Wetterstedt really took lead on when he was a visitor from the Netherlands in my lab, and Sue Trumbor ran some bootleg C14 radiocarbon samples for us, looking at how old is the carbon that we were getting. If you look at the turnover times estimated from biomass from radiocarbon, both on the first cycle and after a number of, I think, 6 wetting and drying cycles, the surface soils date a few hundred years old. The deep soils date 700 to 800 or more years old. So we're mobilizing edible, high-quality substrate that's very old. It's been sitting in those soils for a long time. The question is, how do soils store and stabilize old edible carbon? This is a valuable question for thinking about management. We're thinking about carbon sequestration, because that's really what we want to do. We want to take all this plant carbon, we want to pump it below ground in ways in which it'll stay there. It certainly can happen. I mean, my hypothesis is kind of this, is that in deep soils, the carbon's on the shelf, it's out of reach, the kid can't get to the cupcakes, and so the cupcakes will go stale, even though, my God, they're cupcakes. Or trick-or-treat candy. Come back in a couple of weeks and look at what's sitting in your cabinets. So we've been really interested in these issues of what is driving these processes. How is wetting and moisture and shifts in environmental conditions driving these microbial processes and the flush of carbon and mobilizing carbon and regulating whether or not carbon is stabilized or not? Marcus Kleber hates the word stabilized because we know that the molecules are not inherently stable. We don't believe that anymore, but somehow they're behaving that way, as if they are. Persistent is a nice word in terms of language because it doesn't imply mechanism. Certainly, it fuels microbial growth. Now, this rate, I show some data from just 2 studies as a bit of a side. Sarah Placella, working at our site in Sedgwick, she was a PhD student with Mary Firestone, looked at using ribosomal RNA to look at apparent growth following rewetting. And what she saw was initially very high levels of ribosomal RNA from actinobacteria, then bacilli, and then proteobacteria, which might be a little surprising because we tend to think of the proteos as the fast growers. Postdoc working with Trish Holden and myself, Dr. Michelet, who never unfortunately got this published, had something to do with his trying to bring medical marijuana back from Canada and getting banned from the country, unfortunately. Used BRDU, bromodioxyuridine, which is a thymidine analog. You add it to soils, you incubate it into the soils, it's incorporated into DNA. You can then extract DNA and sequence that DNA and see what organisms actually grew. Other people really since have been using 18O labeled water as a better tool for this because that will label everything, and we know it will, where BRDU may not. And then we analyzed it, and what he saw was initially we see a lot of proteobacteria and almost no actinobacteria. The exact opposite pattern that Sarah did on the same soil from the same site. Different methods, which highlight some of the challenges of doing good microbiology. I think what Sarah really was seeing in the rDNA is that the actinobacteria are just more resistant to drought, and they just carry the ribosomes through the drought. The proteos have to grow, but again, it highlights the challenges of really integrating microbiology and the physics and the whole process work. So she saw actinos, bacilli, then Bacteroidetes and proteos. We saw proteos, Bacteroidetes, and actinos. Hypothesis that actinos hold RNA but reproduce slowly. We could follow that up. It's not something I'm terribly interested in pursuing at the moment, but again, it's sort of looking at that interface between the disciplines, between the scales, that's really challenging to interpret data right. So we decided from what I've shown you is that when multiple drying and rewetting cycles mobilizes carbon that's old and that it mobilizes as much carbon as was in the microbial biomass in the first place, we know it's not microbial carbon that's just being released. A single cycle may release microbial carbon. Multiple cycles have to do something else. They have to be mobilizing soil carbon. And so that had to be probably mineral-associated carbon. So this is a figure from a paper by Andrea Gilling. She's a PhD student with Stuart Grandy. I got to be a junior partner co-author on this one as well. It's really all her work, but it's a very cool paper. Thinking about mineral release, and Eric picked up on this. Eric liked doing field trips, so he went and collected soils from all over California, grasslands from the entire range of sites, surface and deep. You can see the range of soils in the different jars, giving a lovely spectrum of soil color, leaning to the yellows because we're California. And then he looked at the rewetting pulse across all these soils, surface and deep. WEOC is a term, water extractable organic carbon. We use the term WEOC instead of like DOC, dissolved organic carbon, because of course when the soils are dry, it's not dissolved. So we use the WEOC term to be a more precise, this is what we get when we do a water extraction. I don't know what form it was in before we did that. And then looked at the size of the rewetting pulse, and if you look at that across all these soils, you see a few things. And here actually, the pointer will probably be useful. The A-horizon surface soils have higher levels of both, but they all kind of fall along a one-to-one line, just a little below. These pools are a little bigger than the amount of the pulse, but they're closely related, and the A and B horizon soils still fit along a pretty uniform spectrum. The more WEOC, the bigger the pulse. We then looked at microbial biomass using substrate-induced respiration. This is a method where you saturate the microbes with yeast extract, and see how much CO2 they produce. Particularly in B-horizon soils, you can't use chloroform fumigation because the values are just way too low, and you get just disastrously bad data from it. Negative numbers, for example, quite regularly. In this case, the size of the biomass in the B-horizon soils is very low, but the A-horizon soils, we see a very good association between biomass and pulse. So here in the B soils, the amount of substrate that's made available to microbes seems to drive carbon release. For the A-horizon soils, it's better predicted actually by the size of the biomass. More bugs, more CO2. So it's sort of a second-order kinetic relationship in which to respire carbon, you do need microbes, but you also need respirable carbon for them to work with. So it's an added or multiplicative function of them in the A horizon more so than the B. In the B, there's bugs, but it's no relationship. The other thing that Eric found in this is that depending on the nature of the soil, the time course and the relationships were different. So this is looking at B horizon soils, and at our site closest to our home at Sedgwick, if you re-wet, you see a spike, and then that pool of that carbon, the spike, the respiration spike, declines over time. If you go to, and Sedgwick is a smectite mineral-dominated clay loams. They're on fairly alkaline marine sediments, fine-textured clay, messy soils, rich in calcium and other base cations. In Hopland, you get an initial spike that is equivalent to the SIR biomass spike. So that is the maximum rate that we think these microbes can respire at. And then it doubles over the first couple days. So in Hopland, we're seeing this pulse of microbial growth when we re-wet these soils. These are in sandstone, coarser textured, more resource poor, less things to compete for microbes with whatever organics are mobilized. So re-wetting mobilizes carbon. In Hopland, it looks like they can grow. If you look at the pH in water as a really crude index of mineralogy, you see a pattern that the more alkaline soils like Sedgwick tend to show the decline pulse, the more base poor sandy, sandstony type soils seem to show this growth response. So interesting and slightly weird. He wanted to explore this a bit more in terms of what is it that causes carbon to be mobilized in nature. Because the way we tend to do re-wetting studies in the lab is you take soil, you put it in a jar, and you pour water on it. Massively energetic, wildly unrealistic compared to the kind of slow percolative processes that likely occur in the field. And so Eric did this really simple experiment. He took soil cores, incubated some of them basically right side up, and took some of them and just flipped them upside down and put them back in the ground for 18 months. And then came back and then looked at how did just changing the location of sort of B horizon soil affect the mobilization of carbon. How did the nature of natural drying and re-wetting cycles do this? It didn't do anything. So took cores, kept some of them root-free by screening them and checking them periodically, and then came back later and harvested them, using sites around California to get a range of variation in soils and soil types. So there are 4 treatments. There's A horizon soil, which was incubated at the surface. We didn't bother doing A that we buried, which would have been interesting. And then of the B horizon soil, there's stuff that was sampled fresh from at depth. There was stuff that was brought to the surface and kept plant free. And there was soils that were brought to the surface and allowed plants to regrow. And we see some of the same kind of WEOC and SIR versus pulse size graphs here, but in color. And again, you see the same kind of spectrum of behaviors with relatively overall linear responses in which the size of the pulse is both a function of WEOC and a function of SIR. But if we look at these B horizon soils with plants, they're behaving more like A horizon soils. But even the B horizon soils that we incubated without plants, particularly on the microbial biomass side, start behaving a little bit more like planted soils. These values start moving up. We're mobilizing more carbon than are present. It's not super visible in this. But just moving the B soils to the surface increases carbon mobilization. We didn't add carbon. We just somehow are changing the physical dynamics in that system that mobilizes carbon. And of course, injecting root carbon into those systems makes a big difference. We're still really interested in, so what is the nature of that pulse? And what are the different responsibilities of microbial carbon, of the size of the biomass in driving the response to moisture, versus the supply of exogenous carbon or external carbon in doing this? And so Eric did another study in which he measured things continuously, looking at microbial biomass and WEOC every 3 hours, CO2 on Hertz time scales, 1 jar at a time through the erga for several days. When we looked at A horizon, immediately on re-wetting, you get a spike. Okay, there we are. And then it declines. We have already seen that pattern. That's like the Sedgwick pattern. The respiration spikes and decays away. We do see some evidence of microbial growth over the course of that. And if we look at WEOC, the microbes are depleting the WEOC. They're drawing it down. if we look at microbial biomass based on chloroform fumigation, it looks like there's an initial little spike up and then draw down. That may be artifact or it may be real. One of the interesting things, though, is that trehalose, which initially is very high, is consumed almost immediately. And within minutes, you see glucose accumulating. Trehalose is hydrolyzed very quickly. Now, trehalose is probably not so much an osmolyte in terms of stabilizing the water potential, but it's widely used in biology and in biotechnology as a molecule to stabilize proteins against stress. And so what we suspect is going on is that microbes produce trehalose as a mechanism to survive drought stress, not to allow them to remain active, but just prevent their proteins from denaturing. But then they still have to get rid of it to turn back on. And all of a sudden, my God, it's glucose, it's candy. The B horizon soils showed something really different. They showed a classic microbial lag response. So there is a pulse of respiration, but it's delayed by 32 hours or so. There is a growth response. It too is delayed until the microbes turn on. So clearly the microbes were in stationary phase, shut down. There's lots of carbon there, but they're not accessing it. But just re-wetting them was enough to mobilize that carbon and allow them to start growing. But they don't really suck down the WEOC very much, maybe a little bit, but mostly it's unclear where that carbon is coming from. But the biomass is low, but there is a lag. They are so starved, they're not even accessing that carbon. It's a 32-hour pulse, and that time is notable. When we look at the B horizon soil we brought to the surface, we see a hybrid response. It immediately spikes up, and then there's a pulse of growth pulse. You can see the growth pulse again coinciding with the respiration pulse. In this case, the microbes do deplete the WEOC, and the lag is shorter. So just bringing deep soil, to the surface increases the energy inputs and mobilizes carbon and allows microbes to shift their entire growth sort of dynamic. Now, this would be a case where I'd love to have the omics to see who's responding. That wasn't that student, so we don't have that. That would be a next project, but I'd love to do that. So biomass starts higher and grows. WEOC is consumed where it isn't. So we activate the biomass and we get this hybrid. So what have we learned from this? That the overall dynamics are remarkably coherent across the system. We don't see erratic behaviors. We see very describable behavior that we can maybe predict. We can certainly model this sort of kind of second order kinetics in which both the size of the biomass and the available carbon pool both interact to control how much respiration we actually see. And location in the profile really matters a lot, simply because that probably regulates the physical perturbations and the pulsings that mobilize that carbon to microbes. And the way we do re-wetting studies in the lab are therefore probably so artificial that they are probably meaningless in terms of understanding what happens in nature, which is one of those unfortunate lessons, but interesting and valuable. So when we think about water and how it regulates microbial dynamics, in this paper from Sean Schaefer and I, we wrote a number of years ago, if you want to know what happens to carbon, the key thing is understanding access. If carbon becomes available to microbes, it will be eaten. It doesn't matter who eats it, it will be eaten. There are no molecules so inherently chemically stable in soil, or very few, that the ability of microbes to process them is what limits. But what microbes do with that carbon may be fundamentally important in terms of understanding the long-term fate of CO2, of carbon. Does it end up as CO2? Does it end up in polymers? Does it end up in cell-button stuff? Does it end up in dead microbes? Does it end up in exoenzymes? That there's stuff that's going on while soils are dry that I still don't understand. Why do pools of biodegradable, bioavailable carbon accumulate while soils are dry? Our hypotheses for that have all been wrong. It has to be, therefore, I think, something to do with mineral interactions. But why that, I don't know. And a lot of what's regulating this really has to do with the hydrological connectivity and the way water engages and connects microbes with their substrates. that water is not just, as I sort of highlight here, a resource, which is the way we typically think about water unless we're hydrologists. As biologists, we think of water as an essential agent for life. It's also a solvent, and that's one reason why we call it weok. But I also know if I then took those soils and extracted them again, we'd still get more out. There's lots of stuff you can get out that is semi-soluble, sparingly soluble, many other words I might insert there. And it's also a carrier. And then when you integrate all of these together, you get very complicated behaviors that have led us down any number of wrong hypotheses as to what regulates the effects of water on microbes in soil, which is kind of important as soils become drier and weirder. When we think about describing and modeling these to synthesize the mechanistic information up to whole ecosystem scales, we also have to worry about the balance between what I call invisible micromodels like Century, which are very powerful but reach their limits in these cases, and what I might call the too much information models that would have all the microbial gory details in them, which I can probably neither write, parameterize, nor understand, and I certainly can't predict what they're going to do next year because I won't have the microbial data. How do we hit the balance that captures the right level of mechanism? We need something that captures the essential microbial and physical mechanisms to regulate the interactions here. But how do we do that in a way that's parameterizable and understandable? That comes back to kind of this approach. This was this paper that I did with Sean. One approach that came out of a workshop that just published last year, Rose Abramoff was lead on, and came out of a workshop on soil carbon, put together what we called the millennial model. This was kind of a double-edged joke when we named it. We're saying, so if we built Century using our current concepts and measurements of soil organic matter, what would the model look like? So of course, if that was Century, this has to be Millennium or Millennial. Also, it was mostly written by millennials, so it was that's why I got the name. But it looks at these physical fractions of carbon we can measure. Low molecular weight, microbial biomass, mineral-associated particulate and aggregate carbon. Still a first-order model. It can do different things, but it still is a first-order kinetic model. So sort of wrapping up final conclusions as I'm running over time. To really get to where we need to be to understand how soils regulate ecosystems, we really need a sort of a re-energizing of soil science as an integrated discipline. I know too many people who know all the omics but don't know how to look at a soils map because you can't do a soils degree in most places anymore. You can't take those courses as a grad student. But we need to integrate microbes, minerals, and molecules because they do all intertwine with each other, and you cannot understand one without thinking about the others. I have reviewed proposals that were going to solve all the problems of global carbon storage, but the word clay never appeared in the document. So it's like, you may get Nature papers out of all this omics work, but the project will fail, guaranteed. A lot of this does apply to kind of larger scale practical problems. Like if you want to store carbon in soil, well, we better understand better what really regulates it. If we want to manage food production, we need to better understand soils and how to manage them. And we also, therefore, need new generations of models to integrate the insights from all levels of organization, and that an invisible microbe model like Century is not the nightmare, which it would be for some microbiologists who want to see their stuff there. It's the dream. If we can understand the mechanisms well enough to predict their behavior without representing them mathematically and explicitly, that's just all good. But we need to capture the essence of those behaviors in ways that function for what we need to do, and our current generation of tools is not doing that. We're getting there. Thank you very much. I hope we have some time for questions. Sorry. Well, there's the first hand I see.
So I wanted to ask about your second conclusion. You started the talk by talking about the 4 per 1,000 initiative.
Yeah.
And then you walked through a couple of limitations of these steady state models. several of the tools that are used in practice to estimate, say, carbon storage are based on Sentry, like Comet
Right,
Farm.
yeah, to predict, like, if you add carbon, what's the long-term dynamics?
So how do you feel about that?
Skeptical. I love Sentry. I mean, I partly use it as a target because my brother was one of the people who helped develop it, so it's just sort of a fun target. But also Sentry is magic. The power of that model is amazing, but it's being asked to do things that I don't trust it to do reliably. So when you predict the Marine Carbon Project outcome of, you know, a small priming of carbon now will produce long-term benefits, that's for me a hypothesis, not a conclusion. But I think sort of more sophisticated modeling will maybe allow us to improve on that and move it from hypothesis to, okay, I think it's right. But a lot of this is still very speculative. And I also worry about things like nitrous oxide. You add carbon to soil, you make it hold more water. It doesn't take much N2O to counterbalance the climate effect of storing a little carbon.
So we have seen this pulse released during rain event here in New England forest.
Yeah, it's very common.
With Edica variants, right? But there, I think the mechanism might be different. Here, if you remove the O horizon and you start irrigate the soil, you don't see the pulse. And if you if you add O horizon back, you get that pulse.
Okay.
And you take the O horizon, the little layer in the lab, and you incubate. Quickly, the CO2 concentration will saturate your analyzer, right? So here it seems to me that the respired CO2 is actually quite young from the O horizon.
Yeah, well, O horizon, it necessarily is young because O horizon's never radiocarbon old. It's O horizon, organic horizon. There, I suspect there still may be a sort of flush of not used but usable carbon, kind of like our dead root stuff. If it stays dry long enough, small molecules will accumulate, and then you hit them with water, and microbes will blow that stuff off in minutes. I would expect O horizon would look very much like our A horizon, with immediate spike and then decline away. What mobilizes the carbon in that stuff while things are dry, I am less assured of. There may be some enzymatic activity because dry here isn't the same as dry as in California. It may be if there's open photodegradation, certainly plays a role in breaking down surface litter. That's really critical in a lot of grassland systems. Probably not so much in forests, but yeah. These days I have a pretty dry land West Coast bias when I think about these things, and I have not worked in forests in a long time. I'm not good at those.
Maybe this is nice after what you just said. I'm just curious, maybe if there's most of your work is in California and maybe some of the Arctic,
And
right?
Alaska, yeah.
And are there other systems in terms of like creating the next invisible micro model that you think are worth looking at so that model is generalizable to the global scale?
I think plains, grasslands, and ag kind of systems are critical because there's just a lot of carbon in those systems. In terms of soil carbon, grasslands tend to be the biggest storehouse of carbon, for a variety of reasons. That's one reason why Sentry was developed where it was and why it works really well in those kinds of systems.